Okay, next we will welcome up Dr. Chakradhar Mattupalli, Assistant Professor at Washington State University, and his presentation is called Botrytis and Blueberries.
Good morning, all. My name is Chakradhar Mattupalli, and I lead the berry and potato pathology program at Mount Vernon Research and Extension Center. Before I talk about the research, I want to acknowledge the people who made the results possible, and I want to thank all my team members, especially Roshni Baral. She was a master's student, and I'm going to talk about a lot of work that she has done as part of her master's project. I want also to thank Mary, who also worked on some of the screening 2023 botrytis isolates. I want to thank my USDA ARS collaborators and a lot of growers who have allowed us to sample blueberries from their fields. And most importantly, I want to thank Washington Blueberry Commission, Northwest Center for Small Fruit Research, Washington Commission on Integrated Pest Management for funding this several projects that I'll be talking about, and also helping with student success. And also, I want to thank the USDA SCRI program as well as WSDA Specialty Crop Block Grant. This is we recently received funding from WSDA for working on blueberry viral diseases, and this is a project which will be done in collaboration with Chris Benedict and Louis Nottingham. I'll not talk about that project, but the focus of my talk today will be on botrytis. I'll not give a lot of introduction about botrytis because everyone in this room deal with this problem, especially in the blueberry growers, and it's a problem, and currently it is managed through fungicide spray programs involving rotations of several site-specific fungicides, and this pathogen is known is a high-risk pathogen for development of fungicide resistance. So when I started this program like 3 years ago, one of the questions that the stakeholders asked me was to identify what is the current status of botrytis fungicide resistance in the Washington state. So my program is focused mostly on blueberries. But before talking about the research, I want to talk about two currently ongoing projects. This is one of the projects that is currently being done by Mary Steele. She's a PhD student, and the goal of this project was to determine what are the relative susceptible stages for botrytis infection in high bush blueberries. Currently, she's doing a screenhouse. She finished screenhouse studies where she has inoculated blueberry plants at different phenological stages and isolated the pathogen from those inoculated flowers and fruits at different stages. And so the goal with this project is to identify is there any specific phenological stage which is more susceptible to botrytis infection, so we can target the fungicide the most efficacious fungicide at that particular stage. She's giving a talk tomorrow, and her talk is at 9 to 10:30 session. So I would appreciate if you attend that session as well. The second project is a collaborator, a multi-state collaborative project. We call it as a Blue Dynamo, building the latest understanding in extension disease management that yields new and meaningful outputs. I was lucky enough to set up to collaborate with these researchers from different researchers across the country. And the focus of this project is on botrytis fruit rot as well as anthracnose fruit rot. Botrytis is a more problem on the west coast, and anthracnose fruit rot is a problem on the east coast. And the overall goal with this project is to develop effective fruit rot management programs, so we can provide better information to the growers so they can make these effective farm management decisions. So this is a team that we are currently working on. We have researchers from Michigan State. Tim Miles, he's the lead on this project, and we have researchers from Rutgers, University of Maryland, Georgia, and Oregon as well. So there are 4 objectives for this project, and the first objective, what we are working on, is we are trying to characterize the population structure of these fruit rot pathogens. So basically, we would be collecting samples from different blueberry fields across the country, and then we are trying to understand what are the different kinds of pathogens that are associated with the fruit rots. And we would be studying like what kind of a fungicide resistance profiles that these different microbes are there in that are causing the fruit rots. The second objective is to implement the molecular tools for identifying a fungicide resistance. So currently, Mary Steele is a PhD student in my program. She's currently working on developing new molecular tools for identifying the fungicide resistance. So we can easily and detect if there is any fungicide resistance that is present at a field level. And the other aspect with this object is also we also want to understand like the fitness cost which is associated with this fungicide resistance. For example, if an isolate has developed resistance, does it have any fitness cost? Like, does it result in a reduction in the growth or in its ability to produce any spores? So we will study those factors as well. The third objective is about more focused on the horticulture aspects, and the goal is to see like the different maturity and firmness levels and how it impacts the development of a fruit rots. And we are also working on imaging technology to better detect and sort the blueberries that are susceptible to fruit rots. That is an imaging technology which we hope to use it on the packing line. And the fourth objective is putting all this information together and providing it to the growers. And we have recently developed a website where we will be putting all this information that will be generated through this project on this website. Another goal with this project is we are developing a survey in collaboration with the Social and Economic Survey Sciences Research Center. It's an independent third-party evaluator, and as part of this project, SCRI project, we are conducting a survey to better understand the current issues and management of fruit rot diseases in the blueberry fields. So we are doing this survey in all the different states across the US where the blueberries are grown. And the goal with this project is with this survey is to understand like what are the different kinds of management practices that are being followed by the growers and how they are managing these fruit rot diseases. So the results, once we collect all those results, they will be presented in an aggregated form, and they will not be associated with any individual or any contact information. I have these printed questionnaires, but which my team members will be handing over to you. There is an English version and also a Spanish version. I would appreciate if you can fill that paper survey questionnaires. If you are interested in doing an online, please scan this QR code, and the QR codes are also hung up on the screens over there. So if you choose to participate, yeah, you can give and Christina will be giving the Spanish version, and if the other team members will be giving English version. If you choose to participate in this study, you may be eligible to receive a $20 Amazon gift card. because we know how valuable your time and is, and so we really appreciate and thanks for taking the survey. And this gift card will be sent by the Michigan State University researchers upon completion of the survey and authenticity of the responses are verified. So please take a moment to fill up this survey. And before you leave this room, I would appreciate if you return the paper survey back. Moving on, this is the main part. The goal is to understand what is the sensitivity of these botrytis isolates to site-specific fungicides. So the approach that we took was to collect these botrytis isolates from several blueberry fields, and I'll be using the word isolates repeatedly in this talk. And what I mean by isolate is it's a subpopulation of the fungus which is separated from its parent population and maintained in some sort of a controlled manner. So we collected samples at different different stages during the season. We took those samples to the lab. Most of them were asymptomatic, so we surface sanitized those samples. Like these samples included green fruit, mature fruit, and buds, and whatever grew, the fungus, the botrytis grew from those samples. We had maintained them as pure cultures, and we call them as isolates. So in 2022, we sampled 22 Washington fields and 13 fields in Oregon, and we had obtained 376 isolates. And in 2023, we again sampled 22 Washington fields and 5 fields in Oregon, and we obtained 339 isolates. So we have almost screened around like 700 isolates. For this talk, I'll be focusing on the isolates that were screened that were collected in 2022. So how does this test is being done? So it involves like 3 different steps. The first we did an in vitro test, which is based on a fungicide amended media plates. And once we do that, we took a subset of those isolates, and then we did in vivo assays to validate what we saw from the in vitro assays. And finally, we also did detect the mutations in the target gene for FRAX7 group fungicides. I'll talk more about this aspect in a couple of slides. So there are, we screened 10 fungicides belonging to 5 different FRAC groups. These are fenhexamide, fluroxanil, azoxystrobin, paraclostrobin, boscalate, fluoparam, isofetamide, fluxaparaxide, saprodinil, and pyrimethanil. So initially we started doing these assays, and then I want to thank some of the stakeholders who had also suggested me to include premix fungicides such as Pristine, Luna Tranquility, and Switch in the screening. All these fungicides were screened at 1 or 2 doses of fungicide, and we call those as discriminatory dose, which allowed us to differentiate a sensitive isolate from a tolerant isolate. And so in this column, you can see there's different discriminatory doses, and the doses that we used for each fungicide differed, and these doses were based on our preliminary studies as well as the results from other researchers who worked on botrytis in other host systems. Here you see some numbers, and you may be wondering like what these numbers mean to you. So I'll just give one example. I'm sure like many of you are aware of these residue decline curve data. So this is a slide that I got from the US High-Bush Blueberry Council website. And this is a residue decline curve for fluoparam. You can see that this fungicide was detected at a concentration of 1.5 ppm around like 0 days after applying this fungicide, Luna Tranquility. So here, the concentrations that we are testing more or less were comparable to what we are seeing in the field. So I hope this gives you an understanding, like the concentrations that we are testing are at par with what is being observed in the field, because this was one of the questions that was asked previously, is like, oh, what are these concentrations, what these concentrations mean to us. And so hopefully this information would help you align or compare the field and what we are doing in the lab. So for some of the fungicides, you can see, for example, boscalid, the residue decline curve data shows that about 2 ppm of the fungicide was detected in the fruit sample 0 days after application. And in the lab, we were testing at 10 ppm. So it is quite high as what we observe in the field. So how we do these assays? So we spread these the canidia, the spores of these different isolates on a solid agar media, and then observe the growth of these canidia after 16 to 18 hours. So this is how a control plate would look like. Then after that, the isolates are differentiated into 3 different categories, like a sensitive, moderate, and tolerant. A sensitive, basically a sensitive isolate is the one where the spores doesn't grow anything on the media, as you can see here. In the moderate category, you see some growth, but it's about like less than 50% compared to the control. And we considered an isolate as a tolerant if we observed a growth anything about more than 50% compared to the control. So this is our classification. And this classification varied depending on the kind of a FRAC group that we used. So we used this classification for FRAC 7, 11, and 17 fungicides. And for FRAC 9, we used a little different classification, because we used 2 different concentrations, 4 ppm and 25 ppm. And then for FRAC 12, we used 2 categories, a sensitive and then the less sensitive categories. So coming to the results, this is not kind of surprising based on how this fungus behaves in other host systems. We observed that 75% of the isolates exhibited in vitro tolerance or less sensitivity to at least one of the fungicides in the 5 FRAC groups. The other 25% of the isolates that we screened were sensitive to all the fungicides that we used in this study. And the sample size is 376 isolates, and the total number of fields covered were like 22 fields from Washington and 13 fields from Oregon. There were about 11% of the isolates that were screened were tolerant to all the 5 FRAC groups that we tested. If you break up according to each fungicide, here I have a bar graph indicating different fungicides on the x-axis, and on y-axis I have the frequency of the isolates that were tolerant to each of these fungicides. And the gray bars indicate the data from the conventional fields, and the crimson color bars represent data from organic fields. And we had, out of these 22 fields, we had 2 fields from organic and 20 fields were conventional. As expected, isolates from organic fields had lower frequency of isolates with tolerance to the fungicides that we screened, and there were higher percentage of isolates from conventional fields with tolerance to these 10 fungicides. Interestingly, we can see here azoxystrobin and pyraclostrobin, about 80% of the isolates from conventional fields showed tolerance to these 2 fungicides. And regarding FRAC 9, the cyprodenyl and pyrimethamine, even though the percentage of isolates that were tolerant to these 2 fungicides were low, there were about like 40% of the isolates there which are still in the medium category. And Interestingly, these are the FRAC7 fungicides, boscalid, fluopyram, isofetamid, and fluxapyroxide. Boscalid, with boscalid, about 70% of the isolates that we screened, they were tolerant to boscalid. And about 25% of isolates are tolerant to isofetamid. And one more interesting is this fluxapyroxide. Currently, this is fungicide is not registered for use in blueberries in Washington, but still we observed that about 40% of the isolates, they were exhibiting tolerance to this fungicide for which the fungus was not exposed to. This is the data from from Oregon blueberry fields. We had screened isolates from 13 fields, and we were pretty much seeing the similar kind of pattern what we observed with fields from Washington. Higher number of isolates were tolerant to azoxystrobin and pyraclostrobin, and even with the FRAC7 fungicides, you are seeing a pretty higher percentage of isolates with tolerance to these to the FRAC7 fungicides. Coming to the premix fungicides, Luna Tranquility, Switch, and Pristine. Here I have a table which indicates the individual responses to each of these fungicides, pyramethanol and fluopyram, which are the active ingredients in Luna Tranquility. So if we screen these isolates individually to each of these fungicides, for pyramethanol, about 63 isolates were tolerant to pyramethanol and 154 isolates were tolerant to fluopyram. But when we expose these isolates to Luna Tranquility, which has a premix of these two active ingredients, only 14 isolates showed tolerance to Luna Tranquility. We did the same thing for Pristine, which is a combination of a boscalid and pyraclostrobin. And with this Pristine, we observed that about 45% of the isolates that we screened, they were tolerant to Pristine. And with Switch, none of the Botrytis isolates germinated on a 1 ppm fludiaxonil. So our data indicates these are the isolates that were screened screened or obtained from 2022, and our data indicates that currently Luna Tranquility and Switch are still are able to inhibit the growth of the fungus in vitro. So the next thing was like, okay, we did all these assays on a plate, and then the next question is like, why should we believe your results? So we did these in vivo assays. Initially, we started with organic blueberries. We inoculated organic blueberries with this fungus, but we tried thrice, but all the organic blueberries that we received from the store were already infected with botrytis, and so we were able to differentiate the effect of the fungicide from the inoculation that we wanted to do. So we went with using apples as our system. So what we did with this is like we initially poked a hole in the on the fruit, and then we sprayed the fruit with the field rate concentration of Luna Tranquility, Pristine, and Switch. And then we inoculated a subset of we inoculated 17 Botrytis cinerea isolate onto these fruit. And this is again, these different treatments that we had were like an untreated check, which is the first column, which is not sprayed with any fungicide. And so you're seeing a bigger lesion on these fruits. And then the other treatments included Captan, Luna Tranquility plus Captan, Pristine plus Captan, and Switch plus Captan. And again, I want to thank growers who had suggested me that I should include Captan because that's the practice that many of the growers use, is like they mix the site-specific fungicides with a multi-site fungicide. And I really appreciate the grower feedback, and we have incorporated that in our study. And so the take home from this slide is, take home message from this slide is, what we are seeing in vitro is what we are seeing in vivo as well. The first two panels relate to the isolates, to the sensitive isolates, and that's why we are not seeing any lesions when exposed to these different fungicides. But the next two images pertain to Botrytis isolates, which were considered resistant in the in vitro assays, and you are seeing lesions accordingly. I think I have a couple more minutes. And so the first final few slides, I want to show, like, we focused on the SDHI fungicides because those form the backbone in the grower spray programs. What we did, so we had identified several isolates, about 220 isolates, which were resistant or exhibited tolerance to SDHI fungicides in vitro. And so we wanted to understand, like, what are the kinds of mutations that this isolates possess. And having mutations is one of the common ways that the fungus develop resistance to these different fungicides. And one interesting thing with this SDHI7 or FRAC7 group fungicide is that the kind of a mutation that the isolate possesses has an influence on how it interacts with the different fungicides within the FRAC group. For example, if an isolate has a H272R mutation, then the different FRAC7 fungicides that are shown here in ovals, they would be effective. But in this case, if H272R mutation, then all these fungicides would be effective, but boscalid will not be effective. And if an isolate has a H272V mutation and then or P225 mutation, none of the fungicides that are currently available, that FRAC7 fungicide, would be effective. So Tobin Peaver and his team, they did screen several isolates in 2015. And the kind of a population that they were seeing when they did the study was 62% with the H272Y, and then 38% of the population with H272R. So from their study, it seemed like there were more, there were some fungicides which are in the SDHI7 group, which are still effective against botrytis. But in 2022, when we with our data, we showed that there was a reduction in both of these H272R and H272Y. It's only 28%, but the rest, most of the 70% of the isolates have these N230I or P225 or H272V mutation, which means that these isolates would not respond to any of these FRAC7 fungicides, which are currently available for use in botrytis management. This is really alarming, and so growers should pay attention to the population that's being developed, and this is a direct indication of the shift in the population that is happening because of the grower management practices. So this is the key takeaways that I would like you to take from this presentation is that our data indicates that there is a presence of a wide presence of Botrytis cinerea isolates that are tolerant to multiple fungicide chemistries, both from Washington and Oregon blueberry fields. And also we are observing an increase in mutant populations which show cross resistance to different FRAC7 group fungicides. And so with that, I'm happy to take any questions. Yep. Hi, Alan.
Any organic fungicides, what's the chance of developing resistance, or your thoughts on that?
To organic fungicides or the I don't know. I mean, probably I have not screened to any of those, so I can't say for specific. But our data from 2 organic fields that we had collected, they're showing tolerance to these different fungicides that we screened. So I would imagine there would be some level of tolerance to these organic products as well, based on what we are seeing with the conventional products. Alan?
So, I've done efficacy work on the organic fungicides, and one of them, polyoxin D, is a multi-site one, so that's unlikely to develop resistance. The biological products are unlikely to be affected by these resistance mutations. And so I would tell you, most organic products, I think, will be unaffected by these resistance mechanisms, but the trade-off is all these products have low efficacy.
Yeah, sure. I am. So in this image, like the Captain, you can see the second column, that's just with the Captain, which is a multi-site fungicide, and you can still see the pathogen growing on these fruit.
If you wanted to use an organic product that doesn't have these kind of problems, your number one option would be OSO, because that's an organic version of polyoxin D. My question for you is, you have reduced sensitivity in these. Can you equate what you're seeing in the lab with, we haven't really seen at this point, for besides phenhexamide, besides cyprodenil, besides bosclet, we haven't seen field control failures yet, as far as I know. Can you correlate your lab findings to the likelihood we will have control failures for these newer frat group 7 products?
So that's a tricky question. One thing I would like to bring is like the disease triangle. In order to disease to occur, you need to have the pathogen, the environment, and then the host. So we always have the host, and the pathogen is always present based on what we had seen from the past 3 years. We have been collecting samples at different stages, and we are able to collect different isolates, even from the asymptomatic samples. So that means the pathogen is there, the host is there. And in the past 3 years or 4 years that I've been here, it's not been too wet. The environment is not conducive for this fungus to grow. This fungus, that's why this fungus is a lot problematic in the western side compared to the east side of the state. And so maybe that's one of the reasons why we are not seeing the resistance in the field. But as I showed, that the genetics clearly indicates that this fungus possesses those mutations where the active ingredients, especially in the FRAC7, would not work. That's what the data shows from the genetics data and then the in vitro tests. But I think it would be really interesting to see, like, to do some greenhouse studies or do studies in the field where we actually inoculate the plant with these different isolates and then create conducive environment for this fungus to cause the infection, and then do the sprays and then see if they are really effective or not.
Chuck, tell me if you agree with this statement.
Okay.
Even though we have not had conditions conducive for a bad botrytis year, because we're spraying these products, we're selecting for the increased presence of botrytis that have resistance. We haven't had control failures because pressure's not been that high. But when we have a year that's really conducive for botrytis, we're at a high risk of widespread control failures.
Data from 2015, you can see there is population with H272Y and H272R, and then 7 years later, what you are seeing is like it's like 30%, and then the rest 70% is this population with these different mutations. So clearly the biology indicates that the pathogen is causing a shift. And my understanding is that in the past, after 2015, there has been an increase in the use of fluopyram and other new FRAC7 fungicides. And I think that's the reason why you are seeing a shift in the pathogen population. And I believe that it's the the environment should be the factor where it's pretty dry in the past 3 years, and so that's the reason. Yeah, but the pathogen population is there.
So I'm going to give a talk this afternoon that is going to start off where you ended up, and I'll be talking about trying to how to manage resistance when we have the bad year.
Okay, thank you.
Awesome. I think we got one more question.
Yeah, sorry not to interrupt the Alan Schreiber question series there. Thanks a lot for your presentation. I think you raised a bit of an alarm bell, or sounded one at least. I think there was some talk a few years ago about doing a high throughput assay for detecting different isolates. We could be more.
Yes, so we initially started that and we were not successful. We used a plate reader to do that high throughput screening. We were not successful with it, and so we went with the conventional screening. Currently, Mary Steele, PhD student in my program, we are hoping to develop a qPCR, a real-time PCR assay to identify these different mutations. So for these, to generate this data, what we did was we did the sequencing. So to avoid this process of sequencing, we are hoping to just do a PCR-based assay, a molecular-based assay in the lab to do this screening. So that's still in the process. Yeah, and that's a part of the SCRI program that my program is currently working on.
Could you talk a little bit about how that could be used to guide management, practically speaking?
In terms of like the assay or how you can use it. So I think this is the broad goal that I have in my mind is like the growers would submit the samples, like the samples which are actively sporulating, and then we would take those the conidial suspension or the spores from those samples, extract the DNA and run the PCR, and then tell you like, okay, this is the profile of these different mutations that are in your sample. So maybe if your population in your field has more of these H272R, then one recommendation would be like, okay, so you are good to apply any of these fungicides, because as per our current knowledge, if you have population with H272R, only boscalid is not working, but the rest of the other fungicides would be working. So that's the broad goal that I would want to achieve, which would be really, really beneficial for the blueberry growers. So yeah, I'll provide more updates as we make progress on it. All it's on Mary.
Awesome. I think we should wrap up to get these people out of here.
Thank you so much. Yeah, thanks for those questions. Yeah.
Sorry, we're missing a mic back here.
We will be asking for your session feedback by text message. Getting your opinions helps us improve the content each year and helps researchers successfully fund the projects that they're presenting on. We really appreciate your time to respond.
Thanks, y'all. So yeah, we're going to combine times here. So we're going to get those pesticide credit sheets out there in the back. But if you want to wait, while you're waiting in line, you're going to have to wait. If you could take a look at this QR code and the if you prefer by text, or you can just type in that URL, then we can get your feedback and see, because it seemed like people were pretty excited about some of these talks. We'd love to know that you'd like to hear more like this. Another note, just if you're going to head to the next session, which will only be in the other room, the CHS room, you're going to want to head over there pretty quickly after you get your name on our sheets, because I believe that session starts a little bit ahead of 11 a.m. So check your schedules and get over there as soon as you can.